So do you want to start?
Oh, sure. So my name is Mike Prados. I'm a neuro-oncologist, and I've been at University of California, San Francisco for the last 32 or 33 years. And my main interest is in early phase clinical trials, multi-institutional clinical trials, and translational research. And my more recent focus has been on my attempt at precision medicine approaches in children and adults with malignant brain tumors.
So, my name is Santosh Kesari. I'm a neuro-oncologist as well. I am the chairman of the Department of Neurosciences and Neurotherapeutics at the John Wayne Cancer Institute in Santa Monica, California. And my interest is also in precision medicine and precision immunotherapy approaches for brain cancers and trying to develop new drugs for brain cancers as well. One of the things that I think is missed a lot is the fact that most of the drugs we use don't get into the brain very well. Okay. And our goal is really to develop new drugs, and we have a team approach of scientists to do that.
So I'm Lee Hood, president of the Institute for Systems Biology in Seattle and chief science officer for Providence St. Joseph's. And my major interests are applying the tools of systems medicine and big data to the hard problems of disease. And we have a special focus on brain diseases, both neurodegeneration and cancer.
I can bring up some challenges that we face, and certainly in children, which is kind of a major focus for me right now, is one, they're rare diseases, very rare diseases, and genomically fairly bland, as you might know, like brainstem glioma, which I'm going to talk about later. And then access to tissue is challenging, although we do biopsy these children. But then to take that information and find drug cocktails, which we're doing now. We're trying to treat with up to 4 drugs using repositioned as well as positioned drugs. But the issue is that we don't know that any of those drugs get in the brain, and we don't know the sequencing in terms of the PK or the PD hits. And our liquid biomarkers are relatively modest. We can pick up some epigenetic changes in the blood, and we're trying to correlate with outcomes. But it's a very difficult process when we have a rare disease in addition to being in children, in addition to being in the brain, with very little support from the community in terms of pharmacology and industry to help us. I mean, we're doing the best that we can, but it's really difficult. And the disease is already established. We can't. Right now, we don't know of any biomarkers to predict which children get diseases like that. It would be amazing. We do have blood spots in California from all the children that are born, and we're trying to find some predictive biomarker to help us figure out who might end up with these diseases. But the kind of work that you're doing, I think, completely dovetails what we need, which is to find the kids who are going to get it, but then understand what happens once the disease is established. And then how to deal with it with these rare populations. You're looking at larger populations, but each kid is one.
Yeah. Well, I think the nice thing about this dense dynamic data platform is you can do N-of-1 experiments. And you can, and in a sense, the controls, when they're done right, are the individual at an earlier stage before they ever got the disease. Because, again, the arguments I made about not using populations really are true all the way down to the individual level, because unless you're twins, you're going to be quite different from the other person. So I think the whole idea in the future is to get people started on these data cloud analyses so you have the control for what could happen afterwards. And the ultimate in kids is, in principle, we can get to the point where we could follow them all the way through pregnancy.
Right.
And we have to develop more sensitive techniques and different kinds of techniques, but that would be my objective if you really could follow the developing infant and then follow them post-birth and all the way through. Then you'd have really an incredible data set for beginning to look at rare diseases and what they're about and things like that. But I think the other thing that kids present a real challenge on is small blood volumes. So let me just say that what I described today took about 60 mils 3 times. But the point I made is I think we'll have it to the point where we can do all the measurements we need from a drop of blood in 10 years. And even have that be point of care, even at home with a health cell phone kind of thing. So I think we're going in the right direction to give you the kind of data you'll need to really be able to sort through these isolated individual cases and everything.
As a follow-up to that, what are the technologies you're using to look at immune dysfunction in a big data way in patients over time? Because as we are understanding, that is one of the main reasons for developing cancers, as well as when you have an overactive immune system of developing autoimmune diseases.
Well, there is a beautiful panel of 96 cytokines and chemokines and other related molecules that is made commercially available by a company called Olink. And what they need is 2 lambda of blood to make the measurements. It's an incredibly, and the measurements are the most reliable of any of the measurements we make now. They're really beautifully reproducible. And ironically enough, I'm meeting with the CEO of Olink, and I'm going to give him an enormous list of candidates where we'd like to have more assays of this type. And in fact, one of the things I'm really pushing them on are the brain blood-specific proteins, because if you could have a nice ELISA assay where you needed a finger prick to be able to make those measurements, it would be transformational. But we're also looking with immunologist colleagues at a lot of other kinds of measurements of things that we can bring in. I think the immune response is going to be one of the major build-outs that we'll do in the next couple of years to really expand the window through which we see immunity and so forth. And, that result that Charles described of the individual finding cytokines had been perturbed early on, I thought was absolutely fascinating. I guess my question back to you is, if you have any suggestions and thoughts and comments, we're open to adding any of those to our repertoire.
Oh, absolutely. I think we're working on various cellular type of assays in terms of profiling antigen-specific T cells in the blood of patients with cancer right now to define what are their tumor-specific T cells in patients, and why are they working or not working in specific contexts.
Yeah.
And can we use that information to do smarter immunotherapy in the brain context?
Right.
And maybe Mike can comment about immunotherapy and brain cancer and what you think about it and what are the challenges there.
I think we're just at the very nascent kind of stages right now. It's being thrown. It's interesting that you brought up the issue that it's like one size fits all, and it's a little crazy right now.
It is.
It's just a little crazy.
Yeah.
We don't understand enough about any of the biomarkers that relate at least to immunotherapy and brain cancer, and in the individuals, if there are polymorphic, or whatever the profiles are in individuals. And drug companies right now, and we are faced with this dilemma. Drug companies are so fast to move, and they want to get as many patients as they can, right, to find some enrichment group. But they're not thinking about it up front.
Yeah.
And the model systems that we have are a little bit rough around the edges. And we still use model systems. I think patients are probably better, but we still use model systems to try and define what we do. And those certainly haven't been predictive in brain cancer, I'll have to say. And I think we're using a lot of patient resources without a knowledge base first. But at the same time, we are seeing patients in the clinic, and they want therapy. And we want to provide some sense that we're trying to do better. But you know, and as I get older, I get more frustrated because of this lack of base knowledge first.
Yeah.
Right? But at the same time, you know, kids are dying, so you want to do something that you could gain knowledge about.
Yeah.
We're going to do a trial with 96 kids with midline gliomas, brainstem gliomas, using a PD-1 inhibitor and radiation therapy. But we don't know if any of those kids ought to be treated that way. And we're going to collect all kinds of stuff, and it's going to be put into a bucket, and people are going to analyze it without too much of a hypothesis.
Yeah.
And something may come out, and something may not. So it's I wish we could do better.
Yeah.
I really wish we could do better.
See, I think in that trial, one thing that really would be interesting would be to use this cytokine chip and just follow what happens.
Yeah.
And a second thing that would be interesting would be to, if we had an easy way you could analyze the brain-specific blood proteins, because, you know, they map into many, many different brain networks, so it's a very broad view of what
When we
the brain
started
is.
our consortium, we made the decision to be outside of the NCI so that we could take high risk to get those high rewards.
Yeah.
So funding these kinds of
Things is hard.
Yeah, and it's all philanthropy based for us right now in order to do the things that we want to do.
Yeah.
It would be great to have an ability to send blood to your group or something, and to look at liquid biomarkers over time in these kids. So maybe we should talk about that. Maybe we can.
Well, at the very least, it would probably be easy to set something up to do the O-link 96 well cytokine assays and things like that. And cost you almost no blood whatsoever.
Yeah. Well, Lambda, that sounds great. It's wonderful. Yeah.
But and with kids, you obviously can't get that much blood anyway.
Right.
But again, you'd want to make it a longitudinal study.
No, of course.
So you had a sense of change.
Yeah.
And that's what, I mean, so many clinical trials are really not longitudinal in the truest sense. I mean, but you could make that very easily, starting to draw samples before treatment,
they'll all
during
have that.
treatment.
And we'll have tissue blood,
yeah.
I mean, from the tumors as well.
Yeah. So do you want to talk a little bit about the spore that groups at Providence are putting together on brain cancer?
I think one of the unique opportunities and one of the reasons I came to the Providence family of hospitals is because of the large network on the West Coast. I don't remember, is it the third or fourth largest healthcare system in the U.S.? And so tens of thousands of patients with various cancers and in particular brain tumors as well. And so it's a unique opportunity to have a very robust clinical system with actually about at least 4 robust research sites, including the Institute of Systems Biology, John Wayne, Swedish here, and also Providence, Portland. And we're joining forces to really build a unique team of clinicians and scientists. And what we're trying to do is really embrace the complexity and really use systems biology, collect dense data on all of our patients, from the immune functions to the genomics to the proteomics, et cetera, and all the clinical data that's available in systems like EPIC, and really understand and embrace the complexity and try to decipher it and gain insights, and also use it to plan predictive treatment paradigms for patients. Because right now the way we're still doing it is single gene mutations, pick a drug, and that's worked to some extent in other cancers and maybe a little bit in brain tumors on a case-by-case basis. but hasn't really made a difference for the most part in prospective clinical trials. So the hypothesis is that the disease is heterogeneous, the patients are heterogeneous, we need larger data sets, and we need to understand the complexity and use that information.
And we need to be able to do N-of-1 experiments to deconvolute complexity too.
Exactly. And I go back to also, we need better drugs for brain-specific drugs. I think that's one of the challenges, as you alluded to, is and industry really tends to want to, this drug works in lung, let's just try it for brain and XYZ in the same way and same fashion without understanding the underlying biology and science. And that really hasn't helped us in the past few decades.
Yeah. I'm going to have to go.
Oh.
I made an absolute promise I would be somewhere at 10, and I'm just barely going to get there.
Okay.
So is that what you wanted and everything?
Cut off.
Yeah.
Okay.